Now that we've looked at the structure of the membrane, we can talk about how the membrane is used internally inside a cell to separate the cell into specialized compartments. And those compartments are known as organelles. Organelle literally means little organ. So you of course have organs, which are compartments within your body where specialized events occur. Same thing happens in every single cell in your body. You've got these little compartments that will contain special enzymes. They might have a pH that differs from the rest of the cell, etc. We've got these regions where very specialized functions can occur. Now hopefully you remember the names of all of these organelles, and hopefully looking at this diagram here you can name most of them and tell me a little bit about their function. But we're going to go through and look at each of these, and then I'll come back and look at this image again. We have two broad types of cells. We have what are known as eukaryotic cells and then prokaryotic cells. Prokaryotic cells are the bacteria and also a group called the archaea, which look quite a bit like bacteria. They're all little tiny microbes. Prokaryotes don't have a nucleus. That's one of their most notable defining features. There's other features of prokaryotes that define them, but we won't talk about that in this course. We will talk about that in microbiology if you're doing that with me. Prokaryote means before the nucleus. Technically, karyote means kernel or nucleus, pro means before. Eukaryote means true nucleus. EU, if you see that at the beginning of a word, it means true. So eukaryotes have a nucleus and prokaryotes do not. Eukaryotes also have a lot of other specialized organelles that we don't see in prokaryotes. The two eukaryotes you see here are an animal cell and a plant cell. Plant cells are a lot like animal cells. They do everything an animal cell does, and they have mitochondria to break down their food. The difference with plant cells is that they also have chloroplasts to make their own food. So the chloroplast is used to capture the energy from sunlight. That's used to make food, and then the food is broken down in the mitochondrion. Plant cells also have a really tough cell wall around them. That makes up the fiber of the plant matter that you eat. The smallest organelles are quite tiny. Ribosomes are the smallest organelles, and unlike the other organelles, they're not sacs or compartments. They're solid. They're made up of RNA and protein, and their job is to manufacture proteins. But to see a ribosome, you would need an electron microscope. So you can see here what we might look at with an electron microscope, what we could see with the light microscope, and light microscopes are what we use in the laboratory, and what you're capable of seeing with the unaided eye. You can't see any organelles with the unaided eye, and the majority of cells are far too small to see with the unaided eye as well. There are a few exceptions. A frog egg is technically a single cell, and it's quite large. The human egg is the largest cell that humans produce. It's about 1 millimeter in size, so you can see that. If you have the egg on a slide against a dark background, you can just make that out. Note that the scale on the left is a logarithmic scale. And what that means is each step in the scale changes by a factor of 10. So if we start with 1 millimeter here, if we're going up this way, things get 10 times larger with each step. So 1 centimeter is 10 millimeters. And if we go in the other direction, they're getting smaller. So 1 micrometer here, so that little weird U with the tail on it, that is the Greek symbol mu. This is called a micrometer. 1 micrometer is 1/1000th of a millimeter. The other way to say that is 1 millimeter is 1,000 micrometers. Down here we have another unit that you might not have heard of before, and that's the nanometer. 1 nanometer is 1/1000th of a micrometer, or 1/1,000,000th of a millimeter. And you should be familiar with these units. These are units that we use in biology a lot: nanometers, micrometers, and millimeters. The terms cytosol and cytoplasm are quite often confused. They're not quite the same. Cytoplasm refers to everything outside of the nucleus. So everything between the nucleus and the cell membrane, we're including all the organelles, that would be the cytoplasm. The cytosol refers to the actual fluid portion of the cytoplasm. So it's the stuff between the organelles. And it's not entirely water, of course. There's lots of other things mixed in. And we have a lot of fibers that we'll talk about later that are going to help maintain the structure of the cell. These are elements of the cytoskeleton that we'll get to after we look at the organelles themselves. This photograph here is showing the components of the cytoskeleton. So again, the cytoskeleton is made up of long proteins wrapped around each other to form kind of cables that help support all the organelles and keep them in the right place and also help them move around. What they've done to make this image is they've taken a fluorescent dye and attached it to an antibody that will recognize one of the three components of the cytoskeleton. So we have a blue mass there. The dye is attached to what are called intermediate filaments that help support the nucleus. They support the nuclear envelope around the nucleus. And then the green is showing what are called microtubules, which are found mostly in the interior of the cell. They form almost like railway tracks that organelles can move along. And then the orange is showing microfilaments, which are found underneath the membrane and help support the membrane and extensions of the membrane. We'll get back to this. We'll talk about this in detail in part B of this PowerPoint. But part A is going to involve just looking at the organelles. Just a quick overview before we look at each organelle individually. In the center, we've got the nucleus, which of course contains DNA, and that's the genetic information of the cell. We've got an area within that called the Nucleolus, and that's where ribosomes are manufactured. Connected to the nuclear envelope, the membrane of the nucleus, we've got the rough ER, and it is studded with ribosomes. That's the little red dots in this image. Connected to that, we have the smooth ER, which consists of tubes instead of sacs, and it's involved in manufacturing lipids. We have the Golgi complex, which can be used to modify proteins and modify lipids as well, but it's not manufacturing them. And we have the mitochondria, of course, which is responsible for liberating most of the ATP that you use in your cells. And then we've got little vesicles, which are little packets that can contain enzymes, for instance. Lysosomes contain enzymes that are used to break down food that's being taken into food vacuoles. Vacuoles and vesicles can also be used to store stuff, store glycogen or fat, for instance. And vesicles are also used to shuttle material around the cell. Note that of all the phospholipid membrane that makes up a cell, about 50% of it is inside the cell, making up these compartments. So we have about 50% that makes up the surface of the cell, the plasma membrane, and then another 50% that makes up what's known as the endomembrane or endomembranous system. And note that these membranes are connected. So we have the nuclear envelope, which is a double membrane that surrounds the nucleus, and it is continuous with the rough ER, which consists of these sac-like structures, and the rough ER is directly connected to the smooth ER, which consists of these tube-like structures. The rough ER and the smooth ER can bud off little vesicles, and they will contain materials like proteins, for instance, if we're talking about the rough ER. The vesicles can also be used to ship phospholipids around. And these vesicles will be received by the Golgi, so the vesicles will fuse with the Golgi, dump their contents into the Golgi, and then the Golgi can modify those contents. And then the Golgi may produce its own vesicles, and those vesicles could potentially fuse with the plasma membrane, and the contents could get dumped to the outside. Now, one thing to note is the mitochondria are not included in this endomembranous system. So the intermembranous system is very dynamic. It's constantly swapping bits and pieces between the different parts, but not the mitochondria. The mitochondria do their own thing. And it's very interesting why they do their own thing. As we'll see later, they're actually derived from bacteria that became incorporated into our cells. So the mitochondria and the chloroplasts are not considered part of this internal endomembranous system. This is a more accurate depiction of the endomembrane system. Notice that we have all these sacs of the rough ER, and they're all interconnected. and they're connected to the nuclear envelope, and they're connected to the smooth ER, and these sacs are budding off vesicles that are moving around and fusing with other components that make up the endomembranous system. So it's very, very fluid. It's very dynamic. The nuclear envelope is pretty stable. It stays put, and as we'll see, it's supported by elements of the cytoskeleton, but everything else is in constant motion, and materials are constantly being moved around and exchanged. Let's look at each of the organelles in turn, and let's start with the nucleus. So the nucleus is surrounded by the nuclear envelope, and it is a double membrane. So you're not seeing two leaflets there, you're seeing two separate bilayers. There are pores, very large openings, that will connect the interior of the nucleus with the cytosol around the nucleus. And again, also remember that the nuclear envelope is connected directly to the sacs of the rough ER. The lumen, or space, between the two membranes that make up the nuclear envelope can be seen as this dark purple gap. And it's continuous with the lumen found inside the stacks, or sacs, of the rough ER. The image on the left shows a nucleus that has been freeze fractured. We talked about that before. That's where you take a cell and you freeze it very quickly, and then you whack it with a blade and try to split structures within the cell. We looked at freeze fracturing when we talked about the membrane itself, and in those experiments they were trying to split the membrane into two leaflets. That's not what you're seeing here. What you're seeing is a nucleus where the two membranes, entire membranes of the envelope have been pulled apart. We're looking at this with an electron microscope, specifically a scanning electron microscope, which lets us see three-dimensional images of surfaces so we can see textures. And you'll see there's lots of dots and little pits. Those are the nuclear pores. So we've got a nuclear pore here, for instance, and you're seeing that one in section. But these nuclear pores pass entirely through the two membranes. And they are quite large, and they can carry fairly large molecules. In fact, they can carry big chunks of ribosomes, as we'll see. They're also controlled by a little valve-like structure. Of course, the function of the nucleus is to store DNA. A lot of people like to think of the nucleus as the brain of the cell. I don't really like that analogy. I like to use the analogy that it's the library of the cell. So we've got this massive library inside the cell, and the library contains books that we're going to call genes. So DNA contains genes, and each gene codes for a different protein. So imagine you've got a library, lots of books on the shelf. You can pull a book off the shelf and it will give you instructions to make one specific protein. The DNA has to be coiled up to fit inside that space. In fact, if you took all the DNA in a typical human cell and stretched it out and put all the pieces end to end, there would be 2 meters of DNA. And these cells, of course, are not 2 meters, they're tiny. So this DNA has to be coiled up and compacted to fit into that space. DNA is wrapped around proteins. These proteins are called histones. We won't worry too much about that, but it's wrapped around these positively charged proteins called histones. DNA has a slightly negative charge to the strands, so the outside of the strand is negatively charged. It sticks to that protein, and then the proteins allow the DNA to be twisted around and condensed and tightly packed so that it will fit in that space. The DNA becomes even more compacted when the cell is getting ready to divide. So when the cell is getting ready to divide, the nuclear envelope breaks down, the DNA is compacted very, very tightly into individual chromosomes that you can see, and you have 46 chromosomes in most of your cells, except for your gametes, your sperm or egg. There's 23 there, but we'll get to that later. Anyway, you have these individual chromosomes that become very condensed prior to cell replication. And it's proteins that make all this happen. So the DNA is wrapped around these proteins. So if you were to take the DNA out of the cell, you would find that it's wrapped around proteins all the time, even when the cell's not dividing. And we've got basically about half protein and half DNA. And this mixture of half protein and half DNA is known as chromatin. So it's DNA and a bunch of other proteins that ball up the DNA and condense it. And most of those proteins are proteins we know as histones. And just one more look at the surface of the envelope. Down the bottom left there, you can see these pores. And the pores are sometimes referred to as rosettes. They look like a little flower. And each of the petals in the flower is actually a huge collection of many, many different proteins. In the middle of that complex, there is a little plug that can move. It's almost like a plug in a sink. And that plug can move and act like a valve and let things in and out. Now that opening has to be quite large because we have to let ribosomal subunits through. So ribosomes are made up of two parts, two subunits. Those parts are manufactured in the nucleolus, and then they have to leave to get out to the cytoplasm. Messenger RNA has to leave to get out to the cytoplasm as well. The last image there on the right down the bottom shows what's called the nuclear lamina. And the nuclear lamina is made up of part of the cytoskeleton. And you can see we've got these long fibers of protein that are crossing each other, forming kind of a web. And that helps support the nuclear envelope. We don't see that kind of support for the ER. It's far more fluid, but the nucleus is quite a bit more rigid and permanent. Within the nucleus, we have something called the nucleolus. Don't get those two terms mixed up. Students quite often do. The nucleolus is not surrounded by its own membrane. It's just kind of a region. And it's a region that's only apparent if you stain the cells and look at these preserved stained cells. Stains are charged molecules that will stick to proteins and DNA and so on, and they tend to be strongly absorbed by areas where there's lots of activity. There's lots of activity going on in the nucleolus. So here we have the nucleolus as seen under a transmission electron microscope. Very high magnification. Some pretty colors have been added so that we can pick out the different parts. So you can see there's a lot of things going on in the nucleolus because it does stain very darkly. We've got some other areas here that stain quite darkly, and these are areas where the DNA has been very, very condensed with the help of a lot of proteins, and those proteins are staining. We won't get into this, but basically those are areas where DNA has been condensed so much that the genes can't be read. And that's one way that cells will turn off genes. They can turn them off permanently if they're not needed in a certain cell by condensing them and keeping them condensed. But you can also see, of course, the mitochondria in red. Around that, you can see the Endoplasmic Reticulum, which are the little green sacs. So let's take a look at the ribosome, and then we'll come back to the nucleolus when we talk about how it's assembled. What you're seeing here is a very stylized piece of artwork that shows a ribosome bound to some RNA. We've got RNA on the bottom, and the ribosome is moving along the RNA, and it's reading the code that's in that messenger RNA. We have these weird looking things here, which are transfer RNAs, and each one of them is going to carry in an amino acid. And that's what those spheres are up here. And the amino acids are going to be stuck together in a specific sequence based on the code that's in the messenger RNA. And we'll talk about how that works briefly in just a moment. Ribosomes consist of two parts. There's a small subunit and a large subunit, which is larger than the small subunit. Ribosomes can either be floating around free in the cytosol, or they can be attached to rough ER and give it its rough studded appearance. Now, technically speaking, the two halves, the two subunits of the ribosome only come together if there's a messenger RNA that they can bind to. So usually they're floating around as two separate subunits, and then if they come in contact with a messenger RNA, they'll clamp together and clamp on that messenger RNA. It'll travel through the middle. And then they may or may not then move to the ER and stick themselves to the ER and pump the protein they're making into the ER. Ribosomes are remarkably complex structures. As I mentioned before, they're not sacs or compartments like other organelles. They're solid. They're made up of thousands of individual parts, proteins and RNA fragments. The RNA that they're made out of is ribosomal RNA. That's different than messenger RNA. Messenger RNA is going to carry a message from the DNA out to the ribosome. The ribosomal RNAs are used for structure, kind of for scaffolding, and also they're involved in recognizing the messenger RNA and binding to it. And the proteins are there for structure as well, and also to catalyze the reaction that's going to manufacture protein. And here you can see how these parts are put together. So you've got the large subunit at the top, small subunit at the bottom. When they come together, they form a cleft. And as the top subunit rotates, you might see a little green bit. That's where all the magic happens. That's where we have amino acids being brought together to actually build the protein. And of course, that is the role of the ribosome. We've talked about the central dogma of molecular biology before, and let's just revisit that briefly here. So remember that the central dogma of molecular biology states that DNA can be used as a template to make copies of itself, and it can be transcribed to make messenger RNA copies of the information it contains. And then those messenger RNA molecules can be translated at the ribosome to make protein. So DNA contains the information. That library of information is going to remain put in the nucleus. But messenger RNA will carry part of that information, enough information to make one protein, or sometimes a few, and it will carry that information out to the ribosome, and at the ribosome that information will be decoded to make proteins. Remember that proteins do all the work in the cell. So we have DNA within the cell, and it's going to be replicated, but only just prior to cell division. If the cell's not getting ready to divide, the DNA is not going to be replicated. The DNA can also be transcribed to make RNA. And I mentioned this before, but transcription is an English word. If you were to, let's say, read your textbook and then make handwritten notes, you would be transcribing information in the textbook to a page. So transcription, we don't change the language, we're just making a copy. So the RNA is made up of nucleotides. The language, or the alphabet, is nucleotides in both RNA and DNA. So transcription occurs in the nucleus, and then the RNA message is going to move out to ribosomes that might be just floating around in the cytosol, in the cytoplasm. And if that's the case, the protein that's going to be made is going to remain just floating in the cytosol. It's going to be a soluble protein. But the ribosome, after it receives the RNA, might move to the rough ER. And if it moves to the rough ER, then it can pass the protein it's making into the lumen of the ER, or it can embed the protein into the membrane of the ER. After that, vesicles are going to transfer those proteins to the Golgi so they can be modified more, and then those proteins can be secreted so they can be exported, or they can become part of membranes in the cell, including the plasma membrane. That second step, where RNA is using the information it contains to make proteins, is called translation. So I like to think of this as we're going from one language to another language. So we've got that language of nucleotides, and now we're reading that message and we're creating a protein which doesn't use an alphabet of nucleotides, it uses an alphabet of amino acids. Let's take a closer look at those two steps. So the first step, transcription. Again, this is where DNA is going to be used as a template to make a strand of messenger RNA, and this happens in the nucleus. What happens is the DNA molecule is unzipped a little bit at a time, and the information from one of the DNA strands is going to be used to make a complementary strand in RNA. The molecule that does this is an enzyme called RNA polymerase. And just to refresh your memory, there are a few differences between RNA and DNA. First of all, the backbone of RNA contains the sugar ribose instead of deoxyribose. Also, RNA is single-stranded, and that makes it a more delicate molecule. It's not nearly as permanent as DNA. And finally, RNA has the base uracil in place of thymine. RNA polymerase is a complex structure made out of several polypeptides, and it acts kind of like a machine that can run down the DNA molecule and unzip it like a zipper. So the DNA molecule will pass through a groove in RNA polymerase, and it's split into a template strand and a non-template strand. So remember that the hydrogen bonds that hold the nucleotides together are fairly weak. It's not too difficult for RNA polymerase to unzip a few at a time. As the DNA molecule leaves the back of the RNA polymerase, the nucleotides will come back together and the DNA will go back to normal. But while it's unzipped, RNA polymerase will read the template strand and it will add the complementary nucleoside of RNA. So if it sees a T, it will add an A. If it sees a G, it will add a C. If it sees a C, it'll add a G. And if it sees an A, it will add a U. So we get a message that is actually the same as the non-template strand, except that the T's have been replaced by U's, and then that message can go out to the cytoplasm and bind to a ribosome. So at the bottom here, I've got just a short little hypothetical sequence of DNA. This is the template strand. That is going to be used as a template to make the complementary strand in RNA, and that's what's shown at the bottom. Don't worry what the 3' and 5' refer to. They refer to the direction of the DNA molecule and RNA molecule. That's not something we're going to cover just yet. The next step is translation, and that occurs at the ribosome. We've got our message in RNA, and this single-stranded message is going to be decoded by the ribosome into a sequence of amino acids. And the way this works is that the ribosome will read nucleosides in triplets. And each one of those triplets is referred to as a codon. And that 3-letter word codes for a particular amino acid. Remember that we have 20 different amino acids that need to be coded for. With this table, you can decode the message that's found in messenger RNA. And this is universal. And what I mean by that is it works for any organism. So it could be a yeast cell, or a giraffe, or the cell from a fruit fly. Doesn't matter, they all use the same code with very, very few minor alterations in a couple of oddball microbes that we won't get into. But anyway, all organisms use this code, which means you can take DNA from one organism, insert it into another, and that organism will treat that DNA as if it was its own. It can transcribe it and translate it. There are a few codons here that are especially important. One is AUG. AUG codes for the amino acid methionine, but it also tells the ribosome that this is where it should start decoding. It's very important that the ribosomes start at the right place, otherwise we'll get a totally different message. So AUG is always the start of translation. Quite often that methionine is removed later on. And then we have 3 stop codons. So the ribosome will decode the message until it gets to one of these stop codons. The stop codon doesn't code for an amino acid, it just tells the ribosome to stop and to split into its 2 subunits, fall off the messenger RNA and stop doing its thing. Now notice that many of the amino acids you see here have multiple ways to code for them. So proline, for instance, PRO there are 4 different ways to code for it. There's quite a few where there's 2 different ways. There's even a few where there's 6 different ways. So we have this sort of redundancy that's built in. We say that the code is degenerate. Again, that refers to this redundancy that occurs. So the ribosome binds to the messenger RNA and it moves along the messenger RNA to read the code, But this whole system won't work unless we have transfer RNA as well. Transfer RNA will bind to amino acids. What amino acid the transfer RNA binds to depends on the sequence of its anticodon. The anticodon is what binds to the codon. So the ribosome is going to bind to the messenger RNA. It's going to grab onto the appropriate transfer RNA that matches that codon, and that will bring in the appropriate amino acid. Transfer RNA is an unusual molecule. It consists of RNA, but in some places the RNA is double-stranded, as you can see here. So the dots on this diagram represent hydrogen bonds. We've got 3 lobes that are single-stranded, so these 3 loops. And then we've got parts that hold those loops in place that are actually double-stranded. So it kind of looks like a weird 3-leaf clover. Now, just like ribosomal RNA, it doesn't actually code for protein. There are a few other types of RNA that don't code for proteins. We won't be covering those. Just keep in mind that ribosomal RNA, transfer RNA don't code for proteins. Messenger RNA codes for protein. Also in this diagram, you'll notice that some of the nucleosides have a little asterisk on them. Those are nucleosides that aren't the 5 you're familiar with. There are some other ones that are modifications of those 5, but we're not going to get into that either. The key things to note, at the top there we have an amino acid attachment site. So we have 3 nucleosides there. And at the bottom we have 3 nucleosides that form the anticodon. The anticodon will recognize the codon on the messenger RNA. And that particular anticodon will always be associated with a specific amino acid attachment site. And there are 20 different enzymes out there floating around in the cytosol that will grab the appropriate amino acid and then look for the appropriate amino acid attachment site on a transfer RNA and stick it on there. So if everything goes as planned, a particular anticodon will always be associated with the same attachment site, so that a transfer RNA will always bring in the correct amino acid for the codon. Here we can see this in action. So let's take a look at the example up the top there. We have a codon, AGU. We can predict what AGU codes for. We look at our table here and we look at the first base in that sequence. It's an A. We look at the second base, that would be at the top here, and we see that it is a G. And then what we do is we go down to where these intersect, so where the column and the row intersects. That would be in this cell here. The third base in the codon, so this is our third set, is a U. And we see that that intersection tells us this codes for serine. And sure enough, you can see in our example here, serine is the amino acid that is bound to that transfer RNA. I prefer this decoder ring over the table that I just showed you. I think it's a bit easier to use, but it's the same idea. Let's say that we had the codon with the sequence G, A, G. And I apologize for my printing, it's kind of hard to do with a mouse. What we do is we start in the middle and we work our way out. So we have G as our first nucleoside, we have A as our second, and we have G as our third, and that tells us that this codes for the amino acid glutamic acid. Let's look at an example of the entire process in action. So let's say that we have this sequence in our messenger RNA. And this is a very short sequence. It would actually be much longer than this, but just for example purposes. One thing to note is there would be some nucleosides ahead of this and some nucleosides after this, but that's all just junk. That's not going to be decoded. The ribosome knows where to start when it sees that AUG. So what the ribosome will know is that this is how it's going to read it. It's going to read it as a series of 3-letter words. If it didn't know to start at AUG, it might start earlier because there is a preceding message and we'd get something entirely different. But the ribosome will know to break things up into these codons. And then we could use our little decoder ring and we could decode this into methionine, threonine, lysine, glycine. Remember that the start codon AUG codes for methionine. That's the first amino acid in every protein. Quite often the cell doesn't want that to be the case, so quite often it's cut off later, but it always starts out methionine. And then the stop codon is a codon that doesn't actually code for an amino acid. It just tells the ribosome to basically fall off the messenger RNA. Note that we have an N and a C there. That's the N and C terminus of the protein. So if you know the code and you know how the code is arranged, the 5' and 3' refer to specific carbons that are found at the end of the nucleic acid. Again, don't worry about that. But if you know the direction of the code, you also know the orientation and exact sequence of the protein that's going to result. Just a quick refresher before we continue with the endomembranous system after our little side trip. A very important side trip though. I do expect you to be familiar with transcription and translation, just in the level of detail that we mentioned. Recall that we talked about the nucleus. That's number 1 on this diagram. We talked about the nucleolus, number 2. We talked about the nuclear envelope, which is 3, and we've just talked about ribosomes, which are floating around within the cytoplasm, surrounded by cytosol, or they can be attached briefly to the rough ER. So the components we still need to talk about, we need to talk about the rough ER, the smooth ER, the Golgi, and the vesicles that move things around. The vesicles can also move things to the surface, to the plasma membrane, which is number 5 on this diagram. The Endoplasmic Reticulum is by far the largest component of the endomembranous system. In fact, in some cells, it can make up quite a bit more than 50% of the membrane in the entire cell, including the plasma membrane. You can see once again that the sacs of the rough ER have a space within them, the lumen, that's continuous with the space between the 2 membranes of the nuclear envelope. Notice that the rough ER is also continuous with the smooth ER, but they look quite different. The smooth ER doesn't have any ribosomes attached, and also it's tubular instead of being sac-like. Incidentally, the transitional ER here, these are sacs of the rough ER towards the outside that don't have any bound ribosomes, and they're going to give rise to vesicles that will transport proteins off to the Golgi or other places. The diagram at the bottom shows you what the structures of the ER look like in 3 dimensions. The black and white portion shows you what it would look like sectioned and seen under a transmission electron microscope. So the rough ER is going to receive proteins from bound ribosomes, and it's going to modify them. Modifications will include perhaps refolding them, maybe cutting a piece off, or adding sugars, which is something known as glycosylation. After that's been done, typically that material is going to be transported in vesicles, most often to the Golgi, where the proteins can be further modified. And that's going to happen before secretion occurs. The rough ER is also going to be involved in the production of lysosomes, which contain digestive enzymes. The smooth ER doesn't have any bound ribosomes. It's not involved in protein modification. It's involved in the synthesis of lipids. So let's say, for instance, this cell is growing. It's growing in preparation to divide. It needs to expand its volume and expand its plasma membrane. What happens is the smooth ER gets busy and it makes lots of phospholipid, and then those phospholipids are shipped off to the membrane as little vesicles that fuse with the membrane. Another important role of the smooth ER is to detoxify drugs, and it also stores calcium that's used for intracellular communication, events that happen within the cell. A really nice example of this is what happens in muscle cells. Muscle cells have extensive smooth ER. It's modified into something called the Cytoplasmic Reticulum. And don't worry about that yet. We'll talk about that next term. But a sudden release of calcium from the smooth ER in muscle cells is what initiates their contraction. Here you're seeing an extreme close-up taken through a transmission electron microscope that shows ribosomes bound to the endoplasmic reticulum, specifically the rough endoplasmic reticulum. The ribosomes start out floating around in the cytosol. In fact, the diagram's not quite right here because they actually float around as separate subunits. They latch onto a messenger RNA. Depending on the sequence in the messenger RNA, they may just dump that protein into the cytosol, or they may connect to another protein that pulls them to the surface of the endoplasmic reticulum so that they can pass the protein through a little pore in the endoplasmic reticulum. The ribosomes that bind to the endoplasmic reticulum aren't there for very long. They're only there long enough to pass protein into the lumen or into the membrane of the ER. The Golgi body, or Golgi apparatus, was discovered by an Italian neuroscientist named Camillo Golgi. And he had developed some new staining techniques that allowed him to see things that other people had not been able to see. He was also fortunate enough to have some very powerful microscopes that a lot of other researchers didn't have. Amazingly, his discovery was rejected. Everyone thought, people all around the world thought that he had just kind of created this structure artificially. Not that he was lying or anything, but that his staining technique had damaged the cells and created something that's not actually there. That's something known as an artifact. So quite often when you preserve a cell, when you stain a cell, you create structures that weren't there in the cell to begin with. And it wasn't until 1950, which was 52 years after he discovered it, that he was validated and people realized the Golgi does actually exist. That validation came from electron microscopes. So with electron microscopes, we were able to get very good images of Golgi. So it's kind of sad. He died a couple decades before people acknowledged that he was correct. But the Golgi body does exist, and it is a very important organelle. It's part of the endomembranous system. The Golgi body has a noticeable orientation or polarity to it. It has a convex face and it has a concave face. The convex face, so the face that's bended outwards, is the receiving side of the Golgi. This is receiving vesicles packaged with material from the ER. The concave face, the part that's pushed in, is the shipping face, and this is going to ship materials away from the Golgi, perhaps to the plasma membrane, for instance, for secretion, or to other parts of the cell. And here you can see a rather typical Golgi body that's very active, producing large vesicles. And in this case, the receiving face is at the top of the image, and the transport face is at the bottom of the image. The role of the Golgi is to process proteins, sort them, so figure out where they're going to end up in the cell, and in many cases secrete them. So the processing of the protein that occurs is the addition of more sugars in many cases, so glycosylation. That begins in the ER and it continues in the Golgi. The protein may be refolded, so its physical shape may be changed. It's possible that some bits of the protein may be cut off, etc. The proteins will be shuttled to the rest of the cell in vesicles. So they may be shuttled back to other organelles where they're needed. So perhaps there's enzymes that are needed within the ER, for instance, and they'll head back there. Or the proteins could be secreted. So if they're soluble proteins and they're contained within the vesicle that fuses with the surface, then the contents will be dumped to the outside world, or they may end up embedded within the cell membrane. So a protein that is secreted is going to follow the path here. We're going to have ribosomes that will bind to the messenger RNA, And the ribosome is going to move to the rough ER, bind to the rough ER, pass the protein into the lumen of the rough ER. The protein will be modified within the ER and then packaged into transport vesicles that carry it to the Golgi. The protein is going to pass back and forth through those stacks of the Golgi until we have buds coming off of the shipping face. And the buds are going to form vesicles that carry the substance to the surface. The vesicle will fuse with the plasma membrane and the contents will be dumped out. Again, these modified proteins could end up in several different places. They're quite often enzymes, and the enzymes could work in many of the compartments within the cell. So they could go back to the ER, they could go to a lysosome, or perhaps it's something that will be stored in a vacuole. Here's an example of the sort of modification that might occur within the ER and Golgi. What you're seeing here is a carbohydrate. You can see it's made of different parts. So in the legend, you can see that the triangles are glucose, the green is mannose, etc. And what's happening here is this carbohydrate is going to be attached to asparagine, which is one of the 20 amino acids. We start off with it attached to a lipid. Don't worry about that, but this is where the journey for this carbohydrate begins in the ER. So the protein is made by a ribosome, passed into the ER, and then this carbohydrate is pulled off of that lipid molecule and attached to an asparagine. And within the endoplasmic reticulum, it might be modified. It'll be modified again when it reaches the Golgi. So you can see that all those glucose molecules have been ripped off. And on the first part of the Golgi, we have quite a few of the mannose sugars being removed also. In the medial Golgi, we're adding a new sugar, and in the trans Golgi, so now we're towards the shipping phase, additional sugars are being added. And now that protein can leave in a vesicle, and the carbohydrate has been changed quite a bit. And that'll give the protein different properties. One of the important products of the Golgi body is lysosomes. Lysosomes contain digestive enzymes. They also tend to be quite acidic inside, and the digestive enzymes require that acidic environment. This is done for protective purposes. So we talked before about how enzymes tend to have very special needs. The enzymes won't work if the lysosome ruptures. At least they won't if only one lysosome ruptures. If there's some sort of accident in the cell and one of the lysosomes ruptures, there's not going to be enough acid released to change the pH of the cytosol. The digestive enzymes are not going to be active. They're not going to gobble up the whole cell. But if a lysosome fuses with a rather small food vacuole and dumps all its contents into the food vacuole, it will acidify the food vacuole, and those enzymes can get to work on the food that's contained within that little pocket of membrane. Organelles within a cell that become damaged or worn out need to be broken down so that the amino acids, etc., can be recycled. This is also done by lysosomes. So in this case, a worn out mitochondrion has been enveloped in a vacuole or vesicle along with a peroxisome. Peroxisomes are little vesicles within cells that contain additional enzymes and also help bring about chemical reactions that release oxygen and will aid in digestion. So we have the lysosome binding with this vesicle, dumping its contents into the vesicle so that that mitochondria can be broken down and any usable food items can be extracted from it. I mentioned that if one lysosome were to break in a cell, that wouldn't be a big deal because the cytosol is near neutrality. It's not acidic. It's not as acidic as is required by the enzymes inside it. There are a few disorders where people have lysosomes that are a bit too fragile and they break too often, and that causes a great reduction in the quality of life. They tend to be very lethargic and they have quite a few metabolic issues. But there are times when lots of lysosomes will pop suddenly And when they do this to destroy the cell they're in. It's a type of cell suicide. It's known as apoptosis. There are a lot of these mass cell suicides that are programmed into your development. So when you were an embryo, you originally had webbing between your fingers and toes. And of course, that has to go away. So we initially have these webbed hands and feet, and once the fingers start to develop, and we don't need that webbed framework anymore, the webbing between the digits disappears. And you can see that happening here, and it happens quite quickly. So on the left, you can see the hand at 48 days, and then just 3 days later, we've got these distinct fingers. And what has happened is the cells in between the tissue that will become the fingers have undergone apoptosis. Their lysosomes were programmed to burst suddenly and digest and destroy the cells they were in. Let's leave the endomembrane system and take a look at the mitochondrion. So the mitochondrion is separate from the endomembranous system. It does its own thing. It even divides independently of the rest of the cell. It has two membranes to it. So it's got an outer membrane that you can see is smooth, and then an inner membrane that is thrown into all these folds to increase its surface area. Now what's quite fascinating about the mitochondrion is that that outer membrane is very similar to other membranes we see inside eukaryotic cells, has similar proteins and similar composition. But the inner membrane looks a whole lot more like the plasma membrane of a bacterial cell. There are proteins in there that make up these miniature electric motors. And we see these electric motors in bacterial cells, but we don't see them anywhere else in our cells. It's thought that your mitochondria were actually derived from bacterial cells. So prior to 2.5 billion years ago or so, there were no eukaryotic cells. There were bacterial cells, and there were cells called archaean cells. And archaean cells look a lot like bacteria cells. They're prokaryotes, they don't have a nucleus. However, their chemistry is quite similar to eukaryotic cells. And it's thought that an archaean cell engulfed a bacteria cell. And we ended up with two membranes because the innermost membrane came from the bacteria and the outer membrane came from the food vacuole that formed. Now, for some reason, the bacteria wasn't broken down, and there could be a few reasons for that, but one of the most likely is that the bacterium might not have been an unwilling participant. It actually might have been an endoparasite. And there are a number of bacteria that can live inside cells. There are bacteria that infect us. Chlamydia is a good example, that live inside our cells quite happily, and our cells are unable to get rid of them. So it could be that the mitochondria started out as an endoparasitic bacterium, but over time that bacterium became incorporated as an organelle, and these organelles cannot live on their own anymore, but they do reproduce on their own. They reproduce independently of the cells they're in. Now that purple squiggle in the middle of the bacteria cell represents the single chromosome that's found in bacteria. They have a great big circle of DNA and it's all twisted up. Now that didn't entirely go away, so it's not shown here, but there's a little circle of DNA, and that's not to scale, it's quite a bit tinier than that, but there's a little circle of DNA that still exists within all the mitochondria in your cells. It's a circle. We don't see that anywhere else in eukaryotic cells. We don't see it in the nucleus, and it only contains 37 genes, but they're all essential genes. So we have our ancestral prokaryote. Again, this would be an archaean cell. Looks like a bacterium, but the chemistry is similar to eukaryotes. The first thing that would have happened is the plasma membrane would have folded in and given rise to membranes inside the cell. And there are still a lot of bacteria and archaean cells that have internal membranes. But these internal membranes gave rise to the nuclear envelope, the endoplasmic reticulum, the Golgi, etc. Now after that, these cells engulfed bacteria. and they may have been trying to eat them and were unsuccessful. The bacteria may have been endoparasites, but whatever the case, the bacteria became fully incorporated into the cells. They can't live on their own. I mean, in a way, the cell that engulfed them kind of enslaved them. They are an integral part of our cell, and we can't live without them. They produce most of our ATP. Some of these cells went on to engulf cyanobacteria, which are photosynthetic bacteria, and they gave rise to chloroplasts. So chloroplasts are also enslaved bacteria. So we have the two membranes. The outer membrane, again, is smooth. The inner membrane has all of these folds called cristae, which help increase the surface area. And the inner membrane is quite different from the outer membrane. It contains a lot more protein. It's about 3/4 protein. There's a number of important enzymes and other molecules that are embedded in that membrane. The space between the two membranes is known as the intermembrane space, and the space within the very center of the mitochondria is known as the matrix. On the right, what you're seeing is what a rather squashed mitochondria looks like under the electron microscope. Here's another view of a slice through a cell as seen under the transmission electron microscope. You're seeing the longitudinal axis of the mitochondria at the top there. So you're seeing kind of the side view, long view, and then underneath that you're seeing a cross section. Cellular respiration is the series of reactions that liberate energy from your food in every single cell in your body. So your digestive system breaks large molecules up into smaller bits that can be transported to your cells, but it's your cells that get the energy out of those little bits through this series of reactions. The important thing to note is that most of the energy you're able to get from your food comes from reactions that occur within the mitochondria. So the archaean cells were able to do something called glycolysis. If you give them sugar, they can convert that into pyruvate, and they get 2 molecules of ATP out of every molecule of glucose that they start with. So remember, any energy that's liberated in these processes is going to be used to build ATP, and that can shuttle energy to other reactions. But that's all a cell without mitochondria would get out of their food, just the 2 ATP. If that pyruvate moves into the mitochondria will extract another 36 ATP from that one starting molecule of glucose. We're not going to get into this in any great detail in this course. We will in Bio 112, so keep that in mind. But for now, I just want you to realize that the mitochondria are essential for the high metabolic level that we have. If this event hadn't happened where bacteria became incorporated into archaean cells to give rise to eukaryotes, multicellular animals could not have evolved. Multicellular plants could not have evolved. The other thing about the mitochondria is not only is it making ATP, but it's using oxygen to do that. That first step, glycolysis, doesn't require any oxygen. And in fact, most archaean cells and a lot of bacteria cells are poisoned by oxygen. Our cells would be poisoned by oxygen as well if it wasn't for the mitochondria. So the mitochondria came from aerobic bacteria. Those are bacteria that can use oxygen, and they are scrounging up all the oxygen that is found in our cells and in between our cells, and they're using that to produce this ATP. And of course, we evolved a respiration system and a circulatory system to provide all the cells in our body with oxygen. But the oxygen molecules are quite reactive. They're not something that we want hanging out in our cells for too long because they might cause damage, but they end up going to the mitochondria. The mitochondria uses that oxygen to get as much energy as possible out of your food. So how the mitochondria works, just very briefly, again, we have these 2 membranes. We have a space in between them known as the intermembrane space. And what's going to happen is there's going to be reactions that occur within that inner membrane and within the matrix that will pump hydrogen protons out into that intermembrane space. And we'll get this very, very strong electrochemical gradient. So lots and lots of protons between the two membranes. Those protons are trying to get back into the matrix. So they're being repelled from each other because they all have the same charge, and also we have this strong concentration gradient. But the only way that those protons can get back into the center of the cell is through a pore structure that's known as ATP synthase. So here's what's happening. Embedded within that inner membrane, we have all of these proteins that will transport energized electrons that were ripped off of your food molecules. Those energized electrons, as they move from one protein to another, lose a bit of energy, and that energy is used to pump hydrogen protons from the matrix out to the inner membrane space. The reactions that liberate those electrons and pass them on to these proteins occur within the matrix. The hydrogen protons trying to get back into the matrix, the only way they can do that is through this ATP synthase, which, as the name suggests, is going to use that concentration gradient, that electrochemical gradient, to generate ATP. So that last step in the process involves hydrogen protons moving into this really complex structure made out of many proteins that will rotate as the hydrogens pass through. So the hydrogens come into this shaft, and there's this rotor, this cylindrical rotor it's called in this diagram. And the hydrogen protons slot into the side of this rotor, and as a new hydrogen proton comes in, it repels the one that's already there, and it causes this to spin, and it creates mechanical energy that can be used to take ADP and phosphate and slam them together to make ATP. And I realize a lot of this might have gone over your head if you don't remember it just yet. That's okay. I'm not expecting you to know a whole lot about cellular respiration this term. Like I said, we'll get to that next term. But in essence, the mitochondria is a battery. So the mitochondria sets up a voltage or electrochemical gradient across the membrane, and it works just like a battery. These motors, we don't see them anywhere else in our cells. We only see them in the inner membrane of the mitochondria, but we do see them in bacteria. They're used for lots of things. They're used to pump things sometimes. They're used to spin the flagella. The flagella of a bacterium spin instead of waving side to side. So there is quite a lot of evidence to suggest that mitochondria are indeed derived from bacteria. So just in summation, you should be familiar with all these parts of the cell. Now, the one thing I haven't talked about yet that's on this diagram is the microfilament, and that's part of the cytoskeleton. And that's what we're going to look at now in part B. Vesicles and other organelles move around within the cell, but they don't just move around willy-nilly wherever they might. Instead, we have railway tracks that are laid down by the cytoskeleton that guide their movements. We also have cables and pillars that make up part of the cytoskeleton and help hold things in place. The cell is kind of just a bag of water mainly, so it does need some support. Microtubules, microfilaments, and also intermediate filaments are proteins that make up the cytoskeleton. And the cytoskeleton provides mechanical support, especially for very delicate structures like microvilli. But it supports the entire membrane. It provides places for the organelles to anchor to, so it can hold them in place, but it can also direct their movements. It's involved in moving the entire cell. So these components are involved in producing pseudopodia, like a white blood cell would or an amoeba would when it's engulfing things. They're also involved in moving the flagella. Here we can see one of the epithelial cells that lines the interior of the small intestine. This cell has these very thin projections called microvilli that increase the surface area so that this cell can absorb nutrients more quickly. These microvilli are very, very tiny and very delicate. So you can see that they have microfilaments that go up through them to help support them. And there's proteins that will bind those microfilaments to proteins within the membrane. The microfilaments are connected to a web of microfilaments. There is a literal web of these filaments that run through the cell. Towards the nucleus, we also have intermediate filaments, which are larger, and the intermediate filaments will help anchor and support the nuclear envelope. And then also mixed in there, you can see microtubules, which are bigger still, that will help support some of the organelles and work almost like a monorail system for the organelles to travel along. I showed this slide at the beginning of our talk, but it's a really nice depiction of what's happening in a real cell. This is a real cell that was stained with fluorescent dyes that recognize different components of the cytoskeleton. Okay, so there are three types of filaments. The first one here is the microtubule, and the microtubule is quite literally a hollow tube. You can see it's made out of a protein called tubulin, and tubulin comes in two different forms. Those two tubulin molecules will stick together in a pair, and then the pairs are assembled into the microtubule. Microtubules can be added to at one end and taken apart at the other end, and they can be extended and taken apart as needed, so they're dynamic molecules. Next we have microfilaments, and notice that microfilaments are much narrower than the microtubule. And I'm not going to have you memorize those widths, but just know that the actin filaments or microfilaments are the narrowest and the microtubules are the largest. Okay, so actin is again a protein, and the proteins can be stuck together to make this long kind of spiraling thread. But this is a solid fiber, it's not hollow in the middle. And then we have intermediate filaments, and as the name suggests, intermediate filaments are intermediate in size, intermediate in width, between the microtubules and the microfilaments. They can be made up of a bunch of different proteins, but usually it's a protein known as keratin. To demonstrate what the different filaments are doing, I thought I'd use the example of a suspension bridge. This is the Ambassador Bridge. It crosses the Detroit River. It goes from Windsor, Ontario to Detroit, Michigan. That's Windsor in the background there. And kind of an important bridge for me because I worked in Detroit for over 2 years, and I actually lived in Windsor, and I would cross the border every single day, which is about as much of a pain as you would think. In fact, I used to live right about here, kind of like a little troll under the bridge. But anyway, back to the bridge. So there are two important parts to this bridge. We've got a lot of weight that needs to be supported. So the roadbed with the cars on it, extremely heavy. And that weight is supported by cables. And then that force is transmitted to the pillars. So we have our cables shown in white on the bridge. They were constantly painting this thing. So we have our cables here, and they are under a lot of tension. They're resisting stretch, and they're lifting all that weight. And then they're transferring all that weight to these pillars. And all that weight is pushing down on the pillars, all the way down to the riverbed. And these pillars are resisting compression. All that force is being transferred to the pillars, and they are hopefully not going to collapse. They're not going to get smushed. Microtubules, microfilaments, intermediate filaments, they respond to the same sort of forces on a much smaller scale, of course. Microtubules resist compression. Microfilaments resist stretching. And intermediate filaments also resist stretching. Another example would be the old-timey balloon here. So we've got a hot air balloon, and it's within a net. And that net is resisting stretching. So it's providing tension to keep that balloon trapped. And in fact, that network that you see there is a lot like the network of microfilaments that are found under the membrane. So the only difference here is that that net is attached under the membrane in a cell. So microtubules resist compression, and because of that, they can help maintain the cell shape. They play a big role in motility, which is movement. So as we'll see, they play a big role in the structure and movement of the flagella in eukaryotes. They're also involved in chromosome movement. So chromosomes, when they move to either pole during mitosis or meiosis, what they're doing is they're kind of walking along these spindles, the mitotic spindles. And the mitotic spindles are made out of tubulin. They also play a major role in organelle movement. They serve as little railways, basically. Flagella wave from side to side, or at least they do in eukaryotes. They spin in bacteria, but in eukaryotes like ourselves, flagella wave side to side. There's only one cell in the human body that has flagella, and that would be sperm cells. Everyone, though, has cilia, and cilia are basically just small flagella. They have the exact same structure. They wave from side to side. The cilia that you have in your body remain attached to cells that don't move. So when the cilia wave, they move stuff over the surface of the cell. So for instance, there are cilia in the lining of the trachea, the windpipe, and they will move mucus up into your throat so you can swallow it, along with any dirt and dust and bacteria that might have stuck to the mucus. Every flagellum and cilium, no matter what organism we're talking about, paramecia or humans, doesn't matter, the cilia and bacteria all have the exact same structure. And what you're seeing here on the left is a longitudinal section of a cilium. And we're going to look at cross sections from 2 points. The bottom cross section, shown in C, is a cross section of what's called a basal body. These are actually also involved in setting up the mitotic spindle that's used to separate the chromosomes. But in this case, it's embedded into the membrane. It can't move. And you can see that it's made up of triplets of microtubules, 3 microtubules stuck together. The microtubules are all held together by proteins that are shown in blue. Further up, above the plasma membrane, we've got a different arrangement of microtubules. We have 2 microtubules that are found in the middle of the flagellum, and then we have pairs or doublets of microtubules around the outside. Again, the blue represents proteins that are holding this all in place, but the red represents a motor protein that can change shape. And the motor protein is connected between adjacent doublets. Here's what that looks like. So we have this motor protein known as dynein, and dynein can change shape. It can also attach at both ends to microtubules. So let's say we have 2 doublets, pairs of microtubules, and we have dynein molecules between them. If they get ATP, they'll change shape, and that will cause the microtubules to slide past each other. But if they're anchored, so if the base is anchored, they can't slide past each other, and instead they'll bend. They'll bend back and forth, and this is what causes the back and forth movement of a cilium or flagellum. Okay, microfilaments. Microfilaments are much smaller and they are solid. They consist of actin subunits, which are proteins again, that are twisted around each other. They help maintain the cell shape as well, especially the shape of membranes, and they assist in muscle contraction, along with motor proteins that we'll talk more about in just a second. They're involved in cytoplasmic streaming. That's where a cell moves around in an amoeboid fashion. So an amoeba will stick out lobes called pseudopodia, and then it pushes all the cytoplasm into that lobe. And when it does that, what it's constantly doing is liquefying the front part of the cytosol so it can flow into this false foot, and then behind that it's turning the cytosol to gel using these actin polymers. So the cytoskeleton is constantly being reshaped in a cell that's moving around. They're involved in other forms of cell motility, we'll get to that, and they play a role in cell division as well. They're involved in pinching the cell off into 2 parts. So they form a ring, kind of like the drawstring on a pair of sweatpants, in the middle of a cell as it starts to undergo cytokinesis so that it can pinch into two parts. Microfilaments, as I mentioned, also help maintain the cell shape. So you're seeing a scanning electron micrograph here of microvilli that are sticking out from the top of an epithelial cell. And you can see we've got microfilaments that support it, and then they're tied into intermediate filaments that are towards the nucleus. Here's an example of cytoplasmic streaming. So the amoeba that you're seeing there on the right is producing these pseudopodia, and then it's forcing its cytoplasm, forcing its cytosol into those pseudopodia, into those lobes, and that's how it moves forward. What's happening is we have the cytoskeleton being broken down at the front of the cell, and that makes the cytosol very, very fluid. At the same time, we have a gel-like meshwork being built out of microtubules at the back to make it more gel-like back there. And we also have filaments that are sliding past each other to make this movement possible. Motor proteins can also attach to vesicles and other organelles, and then attach to microtubules and move them along the microtubules. So as I mentioned, vesicles have to go to certain places. They're not wandering around aimlessly. Instead, they're following these microtubules. Myosin is the motor protein responsible for muscle contraction. If we look at a muscle cell, it contains these long cylinders called myofibrils. And the myofibrils are made up of structures known as sarcomeres, and they're repeated over and over and over again, end to end. If we look at a sarcomere, it's made up of actin filaments, microfilaments, and that's this here shown in yellow. And also it's made up of a whole lot of myosin motor proteins all stuck together, and that's shown here in purple. Each one of these little bits that's sticking out is a molecule of myosin that can change shape. And what they do is they connect to the microfilaments, and they basically ratchet or walk along the microfilaments and pull the microfilaments closer together. So these microfilaments are going to slide like that. These ones over here are going to slide like that. And what happens as a result is the entire muscle shrinks. And this is the reason that meat contains so much protein. These structures are all made out of protein. Intermediate filaments are made up of long fibrous proteins twisted around each other. Usually these proteins are from the keratin family of proteins. Keratin is the tough protein that you have in your fingernails and in your hair. They help maintain cell shape, especially kind of long-lived structures that aren't all that dynamic. So microfilaments are being torn down and rebuilt all the time because they're associated with the plasma membrane, which quite often changes shape, especially if we're talking about a white blood cell, for instance. So it has to be torn down and rebuilt all the time. The nuclear lamina is the network of intermediate filaments that support the nuclear envelope. And the nuclear envelope doesn't change a whole lot. It's fairly rigid. So unlike the rest of the endomembranous system, it's not all that fluid. So that nuclear lamina will stay there until the cell is just about ready to start dividing, at which case it breaks down so that the chromosomes can be separated. But the intermediate filaments are longer lived. It takes longer for them to be torn down and rebuilt. Again, this image shows the intermediate filaments quite nicely. They're associated with the deep interior of the cell, but they connect to actin filaments that help support structures at the periphery. And here's our terminology list. I did go into a bit more detail than I expect you to know at the moment, so don't worry all that much about cellular respiration. Don't worry all that much about how muscles work just yet, but know the basics, know the importance of the different molecules and organelles that we've talked about.